Hi and Assalamualaikum. We are Diploma in Aquaculture Technology student from Part 3. Welcome to our project presentation of effects on using different media on the algae growth. Hello, my name is Nur Amalina.
My name is Nur Apika Atira.
Hi, and my name is Monica Rina.
This is our table of contents. The objective of this microalgae project is to compare the performance of NPK fertilizer and fish meal in algae growth. Introduction of microalgae. Microalgae is a microscopic and single-cell organism that appear in green color in the water. They use photosynthesis to produce oxygen and reduce carbon dioxide. They also play a crucial role as a food source in aquatic environment, renewable source of biofuel, and treating wastewater.
Introduction of NPK fertilizer in culture. NPK, which is nitrogen, phosphorus, potassium fertilizer in algae culture, can significantly enhance the growth and productivity of algae. NPK fertilizer supply vital nutrients that encourage the growth of algae. In general, the use NPK fertilizer in algae culture promotes the sustainable production of algae for a range in uses such as wastewater treatment and nutritional supplements.
Introduction of fish meal application in algae culture. Fish meal is valuable nutrient source as it provides protein, amino acid and minerals derived from blended trash fish. It supports the microalgae cultivation for better quality of animal feed and biofuels. This organic fish meal can enhance water quality by improved culture medium, but careful management are required to avoid overnutrification. We move on to the methods. Preparation of algae starter. For the materials, we need plastic bottle 500 ml filled with dechlorinated water. Second is solder. Third is anti-chlorine. Fourth is wild microalgae from lake or any water sources. Actually, we should use microalgae powder, but we don't have microalgae powder in the lab. So we just take wild microalgae from UITM lake. For the methods, first we need to make a small hole on the bottle cap by using the solder for oxygen entrance. Second is fill the bottle with 4/5 water and adding 3 drops of antichlorine. Third is put 15 to 20 drops of microalgae taken from the leg in the bottle. Or if we have microalgae powder, we just can use the microalgae powder instead of wild microalgae. Fourth is close the lid and shake the bottle upside down. Fifth, the water bottle is placed near to the sunlight for photosynthesis. Sixth, the water bottle is shaken upside down at least twice a day, either morning or evening. 7. The color of the water will turn green after 1 week, which means the algae already grown. Second method is preparation of fish meal. The materials that we need is dead fish, steel tray, drying oven, blender, and plastic packaging ziplock. For the methods, first, we need to clean the dead fish. Second, the fish were arranged on the steel tray. Third is the oven's fan speed was set to 100% with temperature of 45 degrees. Fourth is the fish were dried for 1 week. Fifth, each day, the fish should be turned upside down to ensure they dry truly. Next, after 1 week, the fish were cooled down and then ground using an electric blender. And the last one is the fish meal was stored in the Ziploc packaging with label. And these are some photos that we took when we do preparation of fish meal.
The third method is counting algal cells. For the materials, we need Neubauer hemocytometer, slides and cover slips, light microscope, and Lugol solution. The method is take a 5 milliliter sample from each culture and place in separate test tubes. Add 1 drop of Lugol solution, mix well. Prepare clean Neubauer slides and covers. Place 1 drop of mixed algae suspension on each Neubauer chamber. Cover with a slip. Let cells settle for 5 minutes before counting. Start counting at the top left square. Count only cells within or touching the chosen boundary lines. For smaller cells, count in 5 smaller squares in the center square. Record the cell count for each sample and plot it on the algae growth curve for each culture. The last method is putting media into the aquaria. Material: plastic aquarium, non-similar size. Air pump, air tubing, air stone, wire extension, white sticking tape, permanent marker pen, NPK fertilizer, 100 grams, fish meal, 100 grams, and light, fluorescent light. The method is: label 9 aquaria as T1R1, T1R2, T1R3 until T3R3. Fill each aquarium with 4/5 of water that has been kept for 2 weeks to avoid chlorine and hard substances. Set up aeration and cover each aquarium with a lid. Add a bottle of pre-prepared microalgae culture to each aquarium. Ensure the aquaria are exposed to sunlight. For the T1, no media added. T2, NPK fertilizer. And for the T3, we add fish meal. After another week, repeat a 20 milliliter sample collection and send the samples to the lab for counting. Record the second set of results in the table. Add 5 grams of NPK fertilizer to each replicate in T2 and add 5 grams of fish meal to each replicate in T3.
Next is results on number algae cells weekly. In this slide, as you can see, there are three different graphs for three different weeks. In this graph, you can see that we use three different treatments to see which one of the treatment is the best for our algae cells blooms. Next is results final numbers of algae cells. The graph shows that treatments 2 NPK fertilizer has the highest productions of algae cells due to the balanced supplies of nitrogens, phosphorus, and potassium required for algae growth. These nutrients support major processes such as proteins productions and energy transfer, as evidenced by Chain's 2018. Treatment 1 is in the second position, supplying some of the nutrients but not in a complete balance like NPK. Treatment 3, which is fish meals, is the poorest because its nutrients are released more slowly and limit immediate algae growth, according to Zhang's 2020. In conclusions, microalgae culturings to compare the performance of NPK fertilizer and fish meals in algae growth involves growing algae under controlled conditions using these two nutrient sources. NPK fertilizer provide essential micronutrients like nitrogen, phosphorus, and potassium, while fish meals offer organic nutrients and proteins. The objective of is to observe and measure the growth rates, cell density, and overall health of the algae under both treatments. This comparison helps determine which nutrient source is more effective for promoting algae growth, providing value insights for aquaculture practice and sustainable algae cultivation.
While working on this microalgae project, we poses many challenges, such as it is challenging to culture the algae starter because the lab does not provide microalgae powder. So we need to rely on wild algae collected from the UIT and lake. So only few of my friends that the algae starter on the water bottle turns green after 1 week. Next is we need to clean the algae culture aquarium at least twice a week due to the presence of insect floating in the water. Next is dissatisfaction and stress often arise during algae cell counting, primarily due to frequent miscommunication. Next is many microscopes were malfunctioning, forcing us to rely on a single microscope to observe microalgae. This significantly slowed down the counting process. Next is many students still struggle with understanding and performing algae cell counting effectively. Lastly, there was miscommunication during the algae calculation, requiring us to start over from the beginning. Here, we attach some of the photo taken while the microalgae project.
Algae culture has many potential benefits for future, such as biofuels, food and feed, environmental benefits, and bioproducts. For biofuels, a potential source of third-generation biofuels is algae. They are a desirable alternative for generating renewable energy due to their high lipid content and quick growth rates. Researchers are refining pre-treatment procedures, harvesting strategies, and growth systems to improve the algae biofuels economy. The second one is food and feed. Production on algae based on food and feed become more popular because of their sustainability and nutritional advantages. They also have high in protein, vitamins, and mineral content. The third one is environmental benefits. Algae are essential for the absorption of carbon dioxide and the treatment of wastewater. They can contribute to reducing the effects of climate change by absorbing carbon dioxide and turning it into biomass. The last one is bioproducts. Important substances like carotenoids, phycobilioprotein, polysaccharides, and omega-3 fatty acids can be found in algae. The food, pharmaceutical, cosmetic, and other industry can use this compound. Algae culture has many implications, including sustainable energy production, enhanced food security, environmental remediation, and health and pharmaceuticals. First, sustainable energy production. By using algae to produce biofuels, renewable and environmentally friendly alternatives are offering to fossil fuels. This action can reduce and help to mitigate climate change. The second one is enhanced food security. Algae are a rich source of essential nutrients including proteins, vitamins, and minerals. Global food security by incorporating algae into the food supply. The third one is environmental remediation. By absorbing carbon dioxide from the atmosphere can helping to combat global warming. Algae can also play a crucial role in environmental cleanup. Algae also can be used in wastewater treatment to remove pollutants and excess nutrients, improving water quality. Last but not least, health and pharmaceuticals. Algae generate bioactive substances with uses in medicine. In order to improve health and wellness, these molecules can be utilized to create novel medications, nutritional supplements, and cosmetics.
For the summary, the general knowledge that my team members and I obtained from this microalgae project is we know that there are specific methods and equipment to count microalgae cells, such as using Newbeyer hemocytometer to count with diameter from 2 to 20 millimeter. Next, we had the opportunity to enhance our problem solving and creative thinking skills by designing a light source for the algae culture. Next, we gain insight into the impact of using NPK fertilizer and fish meal on algae growth. Next, we acquire both knowledge and hands-on experience in culturing microalgae, which is highly beneficial for us as aquaculture students, whether for industry training or future aquaculture businesses. Next, we also learn how to make homemade fish meal, a skill that could help us to produce our own fish meal product in the future. Next is we develop a solid understanding of microalgae cell counting, which deepen our grasp of the concept we study.
Here are the references that we refer on to do on this topic to complete our presentation slides and videos.
That's all from our presentation slide. Thank you.